Alright, this is our online video for Introduction to Cells and their Organelles, or cell parts. Here's your list of objectives. You should be able to explain these when you get to the end of the online video lecture to make sure that you understood everything that was covered. So in starting to talk about cells, we need to look at history, and basically what happened was there were a number of experiments that were conducted, and from this came out the cell theory, and that is one, that all organisms are made of cells. So in order to be considered living, you have to have cells. Next is that all existing cells come from other existing cells. So in other words, presto changeo, it's not like cells just spontaneously show up, out of the universe. They are coming from other cells, and you probably already learned about cell division, mitosis, and meiosis, that would be how we get them from other cells, for example. And the cell is the most basic unit of life. So here we have these 5 gentlemen. They all contributed some piece to this cell theory. And it's really important to note that our understanding of cells really coincided with the invention of the tool to see those cells, and that would be the microscope. When we look at cells, regardless of what cell type you have, whether it's simple or complex, whether it's large or small, all cells generally have DNA. They have some sort of a cell membrane or a boundary going around the outside. They have a cell part that is called a ribosome, and we'll get into an explanation of that in a second. And also they have a gooey substance inside of them, mostly water-based, called cytoplasm. We can divide cell types up into 2 main groups, and that is based on what they contain inside. The first is a prokaryote, and what a prokaryote is it's a type of cell that does not have a nucleus, which is the name of the cell part that holds DNA. So for example, all bacteria are an example of prokaryotic organisms. They are very simple, very reduced in structure, and because of that, they do not have a special nucleus for holding their DNA. Eukaryote cells, or eukaryotic cells, they do have a nucleus and they have other cell parts, and that's going to cover pretty much everything else. So that will be your plants, your animals, and fungi. So let's take a closer look, because it's an important objective to be able to distinguish between prokaryotic and eukaryotic cells. We're going to be, in the second half of this online video, we're going to be looking at all the different functions of these organelles that we're naming. But prokaryotic organisms have no membrane-bound organelles with specialized functions, whereas eukaryotes do. So if you look at this diagram down here, you can see some key differences. Prokaryotes do not have a nucleus, whereas eukaryotes do. You can see that right in the center. And unicellular organisms are only prokaryotic, whereas eukaryotes can be both multicellular and unicellular. So let's just zoom in on these 2 cells here. Alright, and what you're going to see, let me pull my mouse down here for you. Alright, here you can see we have the cell membrane. There are some extra structures here just based on the type of prokaryote this is. But inside you have your cell membrane, or your plasma membrane, that's another word for it. We have our genetic information, that's a ribosome. Okay, and then there is cytoplasm. And that's all that's happening inside the cell. Hop over here and look at all the stuff that's going on in this eukaryotic cell. All sorts of other little cell parts, and these all have their own special functions. And then this right here, this would be our nucleus, and inside this purple stuff here, this is your DNA. Okay, so you can see similarities and you can see some obvious differences. Make sure that you know the differences. So the question is, okay, if there are 2 different kinds of cells, prokaryote and eukaryote, where did they come from? Well, the theory is that prokaryotic cells evolved first. Okay, they're very basic. It's just a membrane, a little bit of DNA inside, some ribosomes to make proteins, but that's about it. So how on earth did we get more complex cells? And this theory is called endosymbiosis, or the endosymbiotic theory. So you sort of have to tell a story in order to do this. And we'll fill in the words here on this page, and then we'll take a closer look at this diagram. So here's the theory. There was a small prokaryote and there was a large prokaryote. So they're both prokaryotic organisms. And what happened was the small prokaryote lived within the large prokaryote, because actually the large prokaryote ate it, intending for it to be made into food. But instead of being digested and broken down for all of its parts, the larger prokaryote found that while inside of the cell and still functioning, the smaller prokaryote provided an advantage. More than likely, that small prokaryote was making proteins that benefited the larger prokaryote, was making excess energy and things like that. So it's sort of like having a little extra piece of the machinery. You know, a good example would be like a V6 engine versus having a V4 engine. Okay, so that little bit of extra energy was really helpful. And so this happened many, many times over many, many generations, okay? And we're talking many years, long, long period of time. And eventually what happened is, instead of the smaller prokaryote being its own living thing inside the large prokaryote, it evolved into just being a part, a non-living part of that larger cell. So let's zero in on this diagram here. All right, let me bring my pointer down here. Oops, there we go. Right, so here we have a large prokaryotic cell, okay, and down here we have a small prokaryotic cell, and this prokaryotic cell used oxygen to produce its energy. So next step, what happens is this small prokaryote is engulfed by the larger prokaryote, and instead of being broken down and digested into pieces, it stays whole, and now it provides an additional function. And this is another type of prokaryote, except instead of using oxygen to produce energy, this prokaryote can perform photosynthesis. And so then this larger prokaryotic cell was able to take in this smaller prokaryotic cell. And what we see in the end here is that that's where they believe the mitochondria and the chloroplast came from, and that those are two cell parts that we're going to be talking about in the second half of this online lecture. So why do they think the mitochondria and the chloroplast were at one time their own prokaryotic living things? Because you look at the data, right? You always need evidence. So here's a diagram comparing bacterial cells to a mitochondria and the chloroplast, and there are some structural similarities. So when we look at the chloroplast and the mitochondria, and we compare them to prokaryotes for today, we see that they all have their own small circular piece of DNA. So it's very unique because other cell parts do not. They all contain their own ribosomes, so they can make their own proteins. So in that way, the mitochondria does not rely on any other part of the cell. It is able to produce everything it needs on its own. They both reproduce in the same manner, and they both have an extra lining or an inner membrane that's very similar in composition to prokaryotic cells. So these are 4 pieces of evidence for why we think mitochondria and chloroplasts were at one time, way, way, way back in history, their own separate living thing that evolved to become a part of a more complex cell, and that's the origin of eukaryotic cells. So what's so special about eukaryotic cells? Because they each contain these or the word right here is organelles, right? These organelles are cell parts that have a special function, and there's definitely a benefit to being able to compartmentalize and have each function of the cell occur in a separate place at a separate time. So we're going to go through them one by one. Make sure you are familiar with their structure and their function. On your mark, get set, go. The cell membrane is also called the plasma membrane, and its main job is it sort of acts like a gatekeeper. It controls what can move in or out of the cell. So even though it looks like it is sort of a solid structure, it's actually porous. So, oops, sorry about that. It's actually porous. So sort of imagine that there are like these little tiny pores or these channels. They're protein channels, actually, that allow materials to either go into the cell or materials to go out of the cell. And so this cell membrane acts as a boundary to separate the cell from the rest of the environment. The cytoplasm, this is something also common to both prokaryote and eukaryote cells, but the cytoplasm is sort of this jelly-like substance, and it fills the interior of the cell, and it provides this nice watery environment to allow chemical reactions to take place more easily. And there will be more on that coming in chapter 2. But we'll get to that when we get back to school in August. So the main job of the cytoplasm is just sort of this watery substance, and it suspends the organelles in place. And you've seen this word quite a bit. Organelle is just a fancy term for cell part, saying that these are small components of the cell, and they all have their own function. The way I like to explain the cytoplasm, imagine you blow up a balloon, okay? The rubbery surface of the balloon is like the cell membrane. Then fill that balloon with a bunch of jello, okay? So it's sort of got a little bit of a solid feel to it, right? It keeps the shape of the water balloon, of the balloon, and then push some marbles into the middle of the jello. What happens? The marbles stick wherever you push them down into. Now, that's sort of what the cytoplasm is like, okay? It's a thicker, jelly, watery substance, and it holds the organelles in their place. Imagine if you did the same thing, but instead filled the balloon with air. So you blow up the balloon with air. What happens when you put the marbles in? Boom. They just fall right to the bottom. So the cell organelles are going to be suspended throughout the 3D shape of the cell. The cytoskeleton, and this is a really neat picture, this is fluorescent microscopy. So we're looking at cells and we're shooting beams of fluorescent light and allowing it to attach onto certain proteins. But the cytoskeleton provides shape and support for the cell. So you can think of the cytoskeleton like the framing of a house. Okay, or like your own skeleton, cytoskeleton meaning the cell's skeleton. Without your bones, your body would just be a pile of goo on the floor. Okay. In addition, there are special parts to the cytoskeleton, and depending on what kind of cell you are, they can form networks of proteins that can help a cell move. So in middle school, you might have learned about some microorganisms that have a special sort of tail that allows them to propel themselves forward, or maybe those short little hairs on the side like the cilia, or just even components inside the cell that are going to be able to flex and contract, and that's going to allow movement like in your muscle cells. So before we move on and talk about any more organelles, the main function of a cell in general, right? What is the cell's purpose? Its purpose is to make proteins, and all of these proteins are going to allow it to carry out whatever function it is that it needs to do for that organism. So there's a group of organelles that all work together to provide this function, and it is called the endomembrane system. The prefix endo there referring to inner. So it's an inner membrane system, not the outside membrane, but the inner membrane. And it's going to be made up of a bunch of organelles that are all connected together, and they're going to help in the producing and the processing of proteins. Making proteins, again, is the most important function of the cell. So I have this diagram at the end of this little section here, and we'll zoom in on it then and take a look at it. So, first part of the endomembrane system is the nucleus, only found in eukaryotic organisms. Stores the DNA, and that helps to keep it protected. Your DNA is like your one copy of that very important instruction manual, and that contains the information or the instructions for how we're going to make proteins. So we keep it inside a nucleus so it's safe from any other sort of harmful things that might be floating around in the cell. Also inside the nucleus, we're going to have a small structure called the nucleolus. I know some of these words are weird, but just get used to them. Try and practice saying them too. And its job is to make ribosomes. And the nuclear membrane is going to wrap around the outside of it, and that's going to help control what moves in and out of a nucleus, just like the cell's membrane controls what moves in and out of the cell altogether. So if we zoom in here, alright, this dense part in the center, this is the nucleolus where the ribosomes are made. The DNA, in this picture, it looks kind of like long, stringy squiggles, and DNA takes different forms or different shapes depending on the artist who is drawing the diagram, and also because during different parts of the cell's life it has different shape as well. And then this dark purple lining, this would be the nuclear membrane. And you can see here when we talk about the endomembrane system, the nuclear membrane is connected to this next part right here, which is going to be another organelle. Ribosomes are very important to the endomembrane system. They are found in both prokaryotes and eukaryotes. They're the only organelle that are common to both. And they are most important because they are the factory workers. They are where we actually produce the protein. And so they are very numerous inside cells. You can find them free floating in the cytoplasm, or in this diagram here, you see how it looks like there's lots of little studs covering this cell part. All those little black dots are the ribosomes. It can attach to what's called the rough ER. So what is the rough ER? Well, the endoplasmic reticulum is what ER stands for, and it's basically a network of highly folded membranes. So we're connecting from the nuclear membrane to the endoplasmic reticulum, more folded membranes. And there are two key types. If you take a quick look at this diagram over here, you see they have different shapes to them. There's sort of this part that's up top here, and then there's a part that's down here. It looks a little more branch-like. The part that's down here is called the smooth ER. It's smooth because it has no ribosomes, so it's got sort of a smooth surface. And that's going to help synthesize lipids, which is a fancy word for fats, and that's also going to help us break down certain toxins like alcohol. The rough ER is studded with ribosomes, and so it looks like it's rough with texture. That's going to help the ribosomes in finishing the protein that was made. So make sure you understand the ribosome is actually the cell organelle that makes the protein, but then the rest of the endomembrane system assists in producing and processing it. Okay, so if we make a protein at a ribosome that's attached to the rough ER, it already has a destination. More than likely, that protein is meant to be secreted outside of the cell, which means we're going to be exporting it. It needs to go to another cell in another part of that organism. Or it's going to be used as a part of the cell membrane. So a cell is constantly building and losing parts of its cell membrane, and again, we'll get into that in a later chapter. But so often it has needs to add more proteins to its cell membrane. The Golgi apparatus is the next organelle that's part of the endomembrane system. So after leaving the ER, we have to package the protein so it can get to the Golgi. And we're going to package it in something known as a vesicle. And a vesicle is basically just a small little sac, and its job is for carrying proteins. So we're going to move from the ER to the Golgi. And once those proteins arrive at the Golgi is going to help sort, package, and deliver the proteins wherever their final destination is. And it's going to use other vesicles in order to move it. So a lot of the times we say the Golgi is sort of like a UPS. So sometimes you can put proteins so that they stay in the Golgi until they need to be shipped out. They can be transported to other organelles in the cell, or they can be sent to the cell membrane to be exported or secreted, was the other word we used on the other slide. So let's take a look at this diagram here. I'd put two big stars on this page right here and right here. This diagram es muy importante, okay? Very important. So the endomembrane system, you can sort of follow the trail here. We start with the information in the DNA, and that information is passed through a nuclear pore. Well, oops, let me come this way. Okay, passed through the nucleus and then goes to a ribosome, let's say this guy right here. And that's where our protein is made. So we read the information that's in our DNA and it produces a protein. Then it's going to move through the ER, okay, as it's being finished. Maybe it moves into some of the smooth ER, it depends. And once it leaves the ER, it goes into this vesicle, okay, and you can see the transport vesicle is going to move it now to the Golgi apparatus. So this is the Golgi here. Alright, and this guy back here, this was the ER. Well, this is the rough ER, so we'll put an R there as well. So our transport vesicle has left the rough ER, has arrived at the Golgi. The protein is packaged, is sorted. It says, here's where I'm going. It goes into another vesicle. Okay, and then this vesicle is going to fuse with the cell membrane, and then those proteins are going to be exported or expelled. And so they're either going to be used maybe as a signaling molecule to talk to another cell, or one of the very many functions of proteins. There are so many. Again, see me in chapter 2. Alright, moving on. So, in order to let all of this happen, we need some energy for the cell. And so, the mitochondria are bean-shaped organelles. They have that double membrane. Remember, we talked about that in the endosymbiotic theory slide. And the main purpose of mitochondria is it converts the food energy that you eat, your glucose or your sugars, and it makes it into usable cellular energy. Okay, and so basically what's going to happen is glucose is going to go into the mitochondria and ATP is what's going to be produced. This is sort of our unit of cellular energy. It's sort of like cell money. It makes everything work. Okay, if you are a special cell like a plant cell or some algae and some bacteria, you then contain a chloroplast because you don't eat food. Instead, you're going to absorb energy from light, and that pigment is going to be called chlorophyll. So the reason why chloroplasts are green is because they contain this green pigment, and it's specifically meant to absorb sunlight energy. So this is found in plants, and they're going to use the energy of the sun to make their food molecules through that process of photosynthesis. Okay, a large sac found in plant cells is called the central vacuole. There might be other small vacuoles that are found in both plant and animal cells to be used as little storage spaces, but we want to focus on this one. This, you can see the arrow here. It's this big thing. It sort of looks like a water balloon, right? This is the central vacuole. It is unique to plant cells. We do not have them in our animal cells. It mostly stores water, but it can store other things like minerals and salts and sometimes some fats and things like that. But basically its job is to help plants keep their structure. So cells can be really floppy because, you know, they're mostly water-based. And so because that water is a liquid and it has a lot of flow, by keeping this vacuole full, okay, it sort of holds the cell as tight and as as shapely as it can. And the best way I can think to explain that is imagine a water balloon inside a shoebox. Okay, that shoebox has a nice sturdy structure, right? And as the water balloon is filled with water, it's going to press against the walls of that shoebox and sort of fill it up. And now that shoebox, instead of being able to flex the sides of it, there's going to be a little bit of pressure pushing back on you. So it helps plants to stand up. That's why if plants don't have enough water, what happens? They look like they're drooping. Okay, their cells they're wilting because they don't have enough water in their central vacuole. Also, to help add some structure, found in plant cells is an additional lining, the cell wall. It's in plants, algae, and fungi, and most bacteria, but it is not in our cells, not in animal cells. The cell wall goes around the outside of the cell membrane, and it provides some additional structure and protection. Okay, if you think about a tree and the bark, okay, and the trunk of the tree and making that wood, all of those plant cells need that really strong cell wall to make it very sturdy. A lysosome, okay, the prefix here, lys, means to burst or break down. Okay, so the lysosome is an organelle that breaks down or destroys things, and this is found in animal cells only. According to current research, there are some arguments about plants. Oops, wrong way. But they contain enzymes, and enzymes are proteins that help with chemical reactions like breaking things down. So what kind of stuff do we need to break down in a cell? We want to break down any foreign invaders. If bacteria enter your cell, you want to be able to break them down so that they can no longer infect other cells. And also, sometimes parts of cells can get old or they can become damaged. So the lysosome is where those old organelles go in order to be broken down so new organelles can be built. And let's just zoom in on this picture down here. Okay, we see a lysosome, and these are all the different things that can come into it. Here's a foreign cell or a particle, something that needs to be digested. Okay, over here we have molecules that need to be destroyed. Maybe we have some viruses that have invaded a cell and we need to get rid of them. Or here we have an organelle and it's received damage in some way and it's no longer functioning. So think of the lysosome sort of as like a garbage / recycling center, because it's going to take in all of these materials, break them back down into their individual cell parts, and then, or not cell parts, I'm sorry, break them back down into little building blocks of things, original atoms and molecules, and that's going to be able to be used to build something else that the cell needs in its place. Centrioles. Okay, centrioles are found in animal cells only, and this is sort of the last organelle that we're really going to talk about here. Their main job is to help the cell during division. So you can see here these centrioles, they're always in pairs, and they have sort of like this log-like structure, so you can see them on either side. But what's going to happen is we're going to organize our DNA, and then we're going to move it to two different sides of the cell. And the idea is you're going to take that one cell, and eventually you're going to split it into two. So you want to make sure there's DNA in each side of the resulting cells, in each of the resulting cells. So the centrioles are going to help with that. Alright, that was sort of like a flash review. Most important thing is being able to understand the structure and the function of each of the organelles in a eukaryotic cell. Go back, check out the objectives. If you have any questions, you can email your teacher or you can ask a question in class.